Hi guys, this is Carol, and this is a mini lecture on patterns of inheritance, chapter 10. So when we talk about genetics, what we use it a lot for is predicting inheritance patterns. And this is some puppies here, and we're looking at their coloration. And so we want to be able to predict. We want to be able to predict the appearance of offspring, for example. Or if there is some genetic problem in a family, we want to be able to predict what is the possibility of it showing up in the next generation. So we know from inheritance, right? We've looked at this before. This is a karyotype. So this would be all of your chromosomes, and you're inheriting. These are not duplicated. Notice how there's only one strand. And you'd get one from your mom, one from dad, one from mom, one from dad, so on and so forth for all of your chromosomes, all of your DNA. Just to be able to really nail this vocabulary, I'm going to go through this. So each gene's locus, it's its location on a chromosome. So when you say, where are your genes? The genes here, for example, here are on this A locus, and there's another gene here, and it's just a sequence of the DNA that is pertaining to a particular gene. Okay? These are duplicated, and we're going to call the pink one from mom and the blue one from dad, and it's already duplicated, and it's got a couple of genes here. A, B, D is being shown. And the other thing that it's showing is it the dominant or is it the recessive, the little d? And so these are different possibilities. Mom here has dominant, so homozygous dominant. Dad has recessive, so it's homozygous also, but homozygous recessive. And the locus is that location, alleles at a particular locus. All this coming out from the nucleus. When we study genetics, we always start out with Mendel, who accidentally was a priest, a father who, behind his church, planted some peas and started to look at, without realizing he was looking at genetics. This is 150 years ago, and he used peas. And why peas? Well, because it was easy. And he noticed that they could be yellow or green, the actual pea itself, or they could be smooth or wrinkled, and they could be short-stemmed or long-stemmed. Or the other easy thing about it is he could pollinate them himself. So he could take one from the yellow and pollinate it onto the green and see what happened. So that was interesting, and he could do it manually without having to rely on an insect. So what caught his attention was why, when you do something like breed a plant that creates yellow peas, and you breed it with one that creates green peas, and you breed those two and you get all of them are yellow. It's like, why was that? Surely some of them should have been green. So he didn't know genetics. He didn't know what was going on. But we can look and say, oh yeah, well, this had the genotype big A, big A. And this had the genotype little a, little a. The phenotype here was green. The phenotype here was yellow. But Mendel wasn't seeing what we now give little letters to understand what's going on. If every single individual inherited one from mom and one from dad here, all of them were going to have at least one big A, making them all yellow. And this is what was so weird to him. It's like, what? Half of them surely should have been green. So he didn't know what to call it, and he called them true breeding plants to those that produced something that was exactly like the parent. So if it was green and it ended up being green, it was true breeding. If it was yellow and ended up being yellow, it was a true breeding plant. That seemed right. Some were not true breeding when he pollinated a yellow with a yellow and he ended up getting some green. And this was what really was a mystery to him. So what he did was actually discover dominant traits. You know, a dominant allele exerts its effect whenever it's present. So you cross a yellow with a green and you'll always get some yellow seeds. So there was dominance, and that's sort of what he first came up with. This terminology, genotype and phenotype. Genotype are the little letters that we're giving it to try and figure out what the alleles are. Phenotype is what it looks like. So he could only see this part. We now can allocate these letters to it, figuring out what dominance. If this is homozygous dominant, it's going to be yellow. If it's heterozygous, I still have one dominant allele, so it's still going to be yellow. And then if I have none of those dominants, it's going to end up being green, and it's called homozygous recessive. So that vocabulary you should know. An important point here is that dominant doesn't mean common. It doesn't mean that. As an example, dwarfism is dominant, but it's rare, and it will only become more prevalent in a population if it confers some sort of advantage to that population. But if it's a trait that doesn't affect anything, it's not going to become more prevalent in a population. So don't confuse dominant with common. Something can be dominant and not very common at all. Okay, this is an example here of in dwarfism. It's little d are the parents of, this is dad here, and of sperm, and then big D, little d, and you would end up with half of them would have that trait of being dwarf. Another piece of vocabulary here is what is meant by wild type and mutant. So wild type is the one that is most common in a population. This is a fly, a fruit fly, and it's the most common form. And now all of a sudden you could have a mutation. Okay, and if you had a weird mutation, and in this case, it's a mutant because it has legs growing out of where the antennae should be growing out. This is some genetic playing with genes, really. That would be a mutant. So if you're not a mutant, you're wild type. So Punnett squares represent gamete formation, as I was showing you before. Let's go really slowly through this one, and then you should be able to understand them. The female is going to be in the egg here, is going to, through meiosis, this is going to divide, and only one of the pair is going to be passed on. So we never know if it's going to be the big Y or little y. For the male who's providing the sperm, again, inherited from his parent, big Y, little y, but when this divides in meiosis, we don't know which is the specific one that will fertilize the egg. So we try every combination and then speak of it in terms of probability. So we put both of these on the square, on the Punnett square. Maybe it's the big Y, maybe it's the little. Maybe it's the big Y, maybe it's the little. And then we fill in the boxes showing what all the possibilities could be. So when we fill this one in, big Y, you would have little y, big Y, still yellow. Big Y, little y, heterozygous. And then little y, it would be green. So the genotypic ratio, how many of these letters do we get? We get one that's homozygous dominant. We get two that are heterozygous and one that is homozygous recessive. Whereas the phenotypic ratio is 3:1. We get 3 yellows to every 1 green. So phenotypic versus genotypic. This is what we call the monohybrid cross. And I have another video where I actually do some of these crosses with you for you to practice. Let's look at one more. It also applies to humans. So in the case of This is cystic fibrosis. And let me get myself out of the way. In the case of cystic fibrosis, it's recessive. It's a recessive inheritance mode. And so the mother here could be, for example, a healthy carrier, big F, little f. The dad, let's say, could be a healthy carrier. So neither of them had the disease. What is the chance of the offspring having the disease? So when we fill the Punnett Square out, we see that they have 25% chance of inheriting that disease because the parents were heterozygous. I'm going to stop it there, and I'll have more on the same chapter.